Bismillah Rahman Rahim 970042 June 24. This is the second video and we'll discuss question 4, 5 and 6 in this video. Bismillah Rahman Rahim. We start with question number 4. Genes have a role in determining the overall phenotype of an organism. Using hemophilia as an example, explain the relationship between a gene, a protein and a phenotype. So they've asked you to use hemophilia as an example. Now you know hemophilia is a sex-linked trait and it is present on the X chromosome. So if a person is X small h, Y, well, that means this male is affected. If the male is X big H, Y, then he's not affected. If the woman is X big H, capital H, then she is not hemophilia. But if she is X big H and a capital H and X small h, then she's a carrier. And this woman, of course, does not exist on the face of this earth because this is a lethal mutation. And a zygote which has this combination will not even grow and will not divide by mitosis and form an embryo and then a fetus. Now, what is it? It's a gene code for a protein which determines the phenotype. So we're talking of the F8 gene. In hemophilia, it's the factor 8 which is not produced. And this is located on the X chromosome. So that's why it's sex-linked. Then it's a recessive allele. And a non-functioning factor 8 protein is produced. So blood does not clot quickly enough, and so there'll be excessive bleeding after an injury. We all have injuries, but then the blood clots. But these people, the blood will not clot. And the fibrinogen will not convert to fibrin. So that is why the blood will not clot, because of the lack of a protein called factor 8. So you can see the wordings of it. A gene codes for a protein which determines phenotype. Then it's the F8 gene. It's located on the X chromosome, and that is why it is called sex-linked. It's a recessive allele. It results in a non-functioning factor 8 protein which is produced. Blood does not clot quickly enough, so there's going to be excessive bleeding, and the person can even die of it. And the reason is that the factor 8 is responsible for the conversion of fibrinogen to fibrin, and the different genotypes and their phenotypes, which I explained earlier to you. You can give that. You had to get your 5 marks. There's a lot of marks for this, 5 marks. So you had to give me a lot of information so I could give you 5 out of 5. In the pea plant, Pisum sativum, the genotype small l, small e results in plants with short stems, dwarf plants. Explain how the L, small e genotype with gene product results in dwarf plants. Now it's very simple. LE is homozygous recessive. So there are two recessive alleles. The small LE codes for a non-functional enzyme. Because it's a recessive trait and because the cause of this is results in short stems. So the some small l, small e codes for a non-functional enzyme. You see this small l, if we wrote big L, small e, then it's fine. Then it's tall stems. Even this results, even if this is results because this got the dominant allele, so this will result in tall stems. But short stems are when both of them are, you have to give me, I have to be very careful in writing it. I don't want to confuse you all. So small l, small e. So if they're both small l, small e, then it's homozygous recessive and it results in short stems. Fine, that is fine. But what does this mean? It means that there is an enzyme which is produced at the end of the gibberellin pathway, in which the last enzyme in the pathway that produces active gibberellin. You see, there is a chemical which is called gibberellic acid or gibberellin, and if that is produced, that results in stem elongation, and so you're going to get tall stems. But if the gene is homozygous recessive, then there will be no cell elongation because no GA will be produced, no gibberellic acid will be produced, so there will be no active gibberellin will not be produced. So active gibberellin will not be produced, so there will not be stem elongation. So you had to give me this for 3 marks. You have to explain this to me why it would result in dwarf plants. Basically, dwarf plants means there'll be short stems. Short stems means something is happening which is not resulting in cell elongation. Now, it's two recessive alleles, homozygous recessive, LE small codes for non-functional enzyme. There's no active gibberellin produced. So no active gibberellin, so no cell elongation. And actually, an enzyme which is responsible in the GA synthesis pathway, there is a GA synthesis pathway from this to this to this, and finally gibberellic acid is produced. Now, the last enzyme, which is GA3B hydroxylase or GA3 oxidase, we don't have to know these names. I'm just explaining it for some of you who want to know the details. And that results in the production of an active gibberellin. Now, that is faulty. That enzyme is not being produced. That enzyme is not working. So if it's not working, so then we're going to have a non-functional enzyme. Even if you have a protein, in that protein, actually what happens, the allele codes for alanine instead of threonine in the enzyme. So one of the amino acids is changed, and that results in the enzyme not having the right shape of the active site. So that is why we say it is a non-functional enzyme. If the active site of an enzyme should be this, and the active site of the enzyme now is this, well, it's not going to function because the substrate is not going to fit. That's why we say it's a non-functional enzyme. Then again, this is something which is given in the book and it is a part of the syllabus. Dwarf plants also occur if the genes controlling growth are not activated, switched on. Explain how DELLA proteins act as repressors, preventing gene expression. So DELLA proteins bind to the PIF. And when it binds to the PIF, what does it do? The PIF is also called the transcription factor. It cannot bind to the DNA, it cannot bind to the promoter. So if it cannot bind to the promoter, then you know RNA polymerase cannot bind to the promoter. So if RNA polymerase cannot bind, there's going to be no transcription. So mRNA is not going to be made. So if mRNA is not made, then how is the protein going to be made? Now all this is again very much from the syllabus, very much given in the book, and everybody needs to revise that and understand that. So DELLA proteins bind to PIF, PIF cannot bind to promoter, RNA polymerase cannot bind to promoter, no transcription, mRNA not made, so the protein will not be made. Question number 5, the gene BRCA1 is expressed in breast tissue and in several other tissues of the body. BRCA1 codes for a tumor suppressor protein. This protein is involved in either repairing damaged DNA or in triggering the death of a cell if DNA cannot be repaired. Some mutations in BRCA1 are associated with an increased risk of developing breast cancer. State one reason why a doctor would recommend that a person has genetic testing for a mutation in the gene BRCA1 and explain why this mutation would put the person at a greater risk of breast cancer. Now, the reason why the doctor would recommend it, if there's a family history of breast cancer, if there is a female and a mother also had breast cancer and a grandmother and a great-grandmother, so it's been coming in 3 or 4 generations, then of course the doctor would recommend that the person has genetic testing. The other factors can be mutation causes changes of primary structure of the tumor suppressor protein. Then changes, folding and binding of the 3D shape of the tertiary structure of the tumor suppressor protein. Then non-functional protein formed. The tumor suppressor protein, if that is non-functional, then of course there's high risk of cancer developing. Then if the DNA is not repaired, because you see they've told you some information in the question and they want you to refer to that. So if the DNA is not repaired or cells with damaged DNA do not die, so this will increase the number of mutations and the mutations will accumulate. Now some mutations cause uncontrolled cell division or uncontrolled mitosis of the breast cells. And that of course results in tumors or cancers, depending on which type of it is. Is it benign or is it malignant? So it was something to do with the question. You were supposed to refer back to the question and tell me things about it. And that is how you were supposed to answer this question. So family history of breast cancer would of course be the top thing, is why a doctor would recommend. And then of course we go into details. Mutation causes changes in the primary structure and then changes the 3D shape of the tumor suppressor protein, and non-functional tumor suppressor protein form. DNA is not repaired, mutations will accumulate, and these mutations can cause uncontrolled cell division. Then coming to the B part of the question, it says DNA sequencing is one method used to test for mutations in BRCA1. Before DNA sequencing occurs, the DNA sample goes through the PCR, the polymerase chain reaction. Suggest why PCR is used for testing for mutations in BRCA1. We only have a small amount of the DNA available. It's one gene. So we want to amplify the DNA to find out if there are those mutations are present or not. So only small amount of DNA available and amplified DNA. That in the UK, 1 in 400 females have a mutated allele of BRCA1. 70% of females with the mutated allele of BRCA1 will develop breast cancer by the time they are 80 years old. In 2022, the population of females in the UK was 3.5 into 10 raised power 7. Calculate the number of females in the UK population in 2022 who it is estimated will develop breast cancer by the age of 80 years old. Give your answer to in the standard form. Show your working. So this is a little bit of maths. 1 in 400 females have mutated. So then you have to divide 3.5 into 10 raised power 7 divided by 400 and you got a figure. And then 70% of that figure. And then it said give your answer in the standard form. So you have to give it to me in this form, which is 6.1 into 10 raised to the power 4. Or you could have said 6.125 into 10 raised to the power 4. So this would get you your 2 marks because you'd have done the calculations of 1 in 400 females out of this, and then 70% of those females. So this was a little bit of maths which we had to do. Then the part D of the question is one advantage of genetic screening is to determine which drugs should be selected to treat cancer. A study was carried out on women with breast cancer who had been treated with DNA damaging drug that kills tumor cells. The women had all been tested for BRCA1 mutations. One group had a BRCA1 mutation and the other group did not have a BRCA1 mutation. The researchers used data to assess the probability of survival for the women in each group of the study after treatment of the drug. Figure 5.1 shows the probability of survival over a time period of up to 180 months. And then you can see probability of survival, and then you can see group with BRCA1 mutation and group with no BRCA1 and probability of survival, how it is changing, and the months that were, it was a study done over 180 months. So this was a group with no, and this is all the data which is given. Then it says the researchers suggested the hypothesis that the DNA damaging drug is more effective in women with BRCA1 mutation. Discuss whether the results in figure 5.1 support the hypothesis suggested by the researcher. So I expect you to really tell us from the graph what was happening. So the women with BRCA1, let's look at the graph. Woman with BRCA1 is this one. So the probability of survival is 1 and over a period of time it is greatly decreased. But the women with no BRCA1, it has in fact a probability of survival has decreased more. So women with BRCA1 have a higher probability of survival. Probability of survival was higher in BRCA1 and the probability of survival decreased less. The probability of survival decreased less for the women with BRCA1. And then you could give me a data quote that at 30 months the data survival with mutation was 0.975 and without mutation was 0.85. So any data quote which you could have given me in the mark scheme, of course, there's a whole list of the data quote and everybody should go through that. And then of course when it says suggest, hypothesis, more effective, then you say study carried out for a very long time, 15 years. And why does it not support is because there's no statistical test carried out. And then we don't know what was the sample size. So those are the things which we write in does not support because it says suggest more effective than this or so the thing is because you see discuss whether results support. When it says support, then you must remember to give me something that says it does not support. So there were only 2 marks for it, but of course then you need to get these 2 marks. So women with BRCA1 higher probability of survival, women with BRCA1 probability survival decreases less, and study carried out for 15 years, no statistical test carried out. That is of course the one which does not support this hypothesis. And then the data quote, this is the data quote. The data quote is that at any, you could have given me anything, I'm just giving you one of them. You could have given it 60 months, you could have given it at 90, 120, 150, 180. So the 30 months with mutation was 0.975 probability of survival, and without mutation it was 0.85. So any of these points you could give me and you would get 1 mark for that. Now coming to question number 6, figure 6.1 is a diagram of a mitochondrion, diagram of part of a mitochondrion. This is the cytoplasm, this is the intermembrane space, this is the outer membrane, and then this is the inner membrane. Now it says on figure 6.1, use arrows to show the route taken by oxygen from the cytoplasm into the mitochondrial matrix. Well, very simply, it's going to move from the cytoplasm through the phospholipid bilayer. It's a small uncharged molecule, so it can pass through the phospholipid bilayer. But if you show it through any of the channel proteins, that is a reject. You won't get this 1 mark. It is going to pass, the oxygen molecule can pass through the phospholipid bilayer, the outer membrane and the inner membrane, and will enter the matrix. So the arrow would have been right through it, but not through any of the channel proteins. You would lose the mark for that. Then it says identify protein D and the group of proteins represented by A, B, and C and describe their roles in oxidative phosphorylation. Now, this of course is a long question. It's for about 7 marks. Wow. So identify protein D. What was protein D? Let's look at protein D. That's ATP synthase. ATP synthase. So ATP synthase would get you a mark. A, B, and C are the electron transport chain, or you can say the electron carriers. And then, of course, electrons traveled along ETC, energy released to pump actively hydrogen ions or protons into the intermembrane space. So then we're going to have the hydrogen ions pumped into the intermembrane space here. And so electrons are going to move along the ETC. Electrons are going to move along the ETC. Electron transport chain, these are the ones, the electron transport, not the whole of it. So the electron carriers would be these. And energy release, pump protons into the intermembrane space. Now this will increase the concentration of hydrogen ions here. So more hydrogen ions, this will create a proton gradient, and the protons will diffuse through the ATP synthase. And when they diffuse through the ATP synthase to the matrix, synthesis of ATP from ADP and inorganic phosphate, and this of course is called chemiosmosis. So a very direct question, a very easy question. Everybody should have figured this out. Identify protein D and the group of proteins represented by A, B, and C and describe their role in oxidative phosphorylation. So a lot of points available for you. D is equal to ATP synthase, A, B, C is ETC, electrons travel along ETC, energy released, pump hydrogen ions or protons into the intermembrane space, creates a proton gradient. H positive diffuses through ATP synthase to the matrix, synthesis of ATP from ADP and inorganic phosphate. This Pi is inorganic phosphate, and this process is called chemiosmosis. So 10 points. These two, of course, were essential. Any 5 out of these 8. Then we come to part C of the question. The matrix of mitochondria contains small circular DNA. In the cells of humans, the mitochondria have circular DNA that contain 37 genes. Suggest functions for these mitochondrial genes. Named enzyme for the Krebs cycle. If you said maybe an enzyme which you are used in the Krebs cycle or the link reaction, dehydrogenase, decarboxylase. Then ETC proteins, electron carrier proteins A, B, and C. Then the ATP synthase, which is D, is coded by a gene which would be for ATP synthase. Then membrane or channel proteins or transport proteins. And then the DNA, which makes the tRNA and the rRNA or the ribosomes. So a lot of answers were available for you, and you had to just be a little imaginative and know your bio well. So enzymes for Krebs cycle, link reaction, ETC proteins or the electron carriers, then the gene for ATP synthase, then the gene for transport and carrier proteins, and then gene to make tRNA or ribosomal RNA or ribosomes, which are actually needed. RNA is needed for the ribosomes. Question number 7. Student investigates the effect of temperature on the rate of photosynthesis at two different light intensities. The rate of photosynthesis is determined by measuring the rate of oxygen production of the plant. Figure 7.1 shows the results of the investigation. So there's one at low light intensity and then one is at high light intensity. The rate of oxygen production is millimeter cube per hour. At low light intensity is the same as very little. The rate of oxygen production is less than 0.5. And in high light intensity, of course, at 10 degrees Celsius, it is 1, and then it keeps on increasing, and then it increases till 30 degrees Celsius and then goes down and the rate starts to decrease. The rate starts to decrease. Now, with reference to figure 7.1, describe and explain the results of the investigation for the plant in low light intensity. So rate of oxygen production is low or stays constant. Light intensity was the limiting factor because it's low light intensity. So light intensity was the limiting factor. So less light absorbed, less photolysis, less light dependent reaction. So you have to understand that because low light intensity, you couldn't have said no light energy absorbed. Some of it is being absorbed, and there is the rate of oxygen production is there is less than 0.5, but it is there. So with reference to 7.1, describe and explain. So rate of oxygen produced stays low or is constant, or you could have said small decrease in rate of oxygen produced or rate of oxygen produced or rate of photosynthesis. There is a small decrease. You see, you can see the line. It is here, and then it is decreasing a bit. So that would be a describe. And the explanation would be light intensity limiting factor, less light absorbed, less photolysis, or less, you can say, the less light dependent reaction. So rate of oxygen produced is low. This will be the describe. Explain would be these. This would be the describe. So less light intensity was the limiting factor, less light energy, less photolysis, less light dependent reaction. And the part 2 of the question says, with reference to figure 7.1, describe and explain the results of the investigation for the plant in high light intensity above 30 degrees Celsius. So let's look at the graph again. What is the graph in above 30 degrees Celsius? Now, above 30 degrees Celsius, this is the thing, that the rate is decreasing. This is 30 degrees Celsius. So rate of oxygen produced is decreased. You got a mark for that. Then you gave me paired data. At 30 degrees Celsius, the rate of oxygen is 4 millimeter cube per hour. At 50 degrees Celsius, it is 0.65, 0.65, or anything between 0.62 to 0.65 was acceptable. And you gave me this data, you got a mark for that. Why? Because enzyme ATP synthase is denatured. You could have said that ATP synthase or NADP reductase is denatured, and there's going to be a change in active sites, so fewer enzyme substrate complexes formed. A very simple explanation for that, not very difficult explanation. So 30 degrees, the rate is decreasing. So basically, something's happening to the enzyme which is responsible for this oxygen production. So rate of oxygen produced decrease. Rate of oxygen produced decrease 30 degrees Celsius, 4 millimeter cube per hour. ATP synthase denatured, change in active site, fewer enzyme substrate complexes formed. Then coming to the B part of the question, very direct question, 3 marks. Describe the process that produces oxygen during non-cyclic photophosphorylation. Non-cyclic photophosphorylation, light absorbed by photosystem 2 or P680, which results in photoactivation of the chlorophyll. And the second mark scheme point is photolysis of water. Number 3 mark scheme point is water splitting enzyme. And the number 4 mark scheme is water splits into protons, electrons, and oxygen. Water splits into protons, electrons, and oxygen. Or you could give me the equation for it. So light absorbed by photosystem 2, photolysis of water, water splitting enzyme, water splits into proton, electrons, and oxygen. Well, you could have given me the equation for it as well, which I've also given you here. And so you could have given me that. That would be acceptable as well. So water splits into 2H plus 2 electrons and half O2. And of course, then another half O2 combines to form one oxygen molecule. So that completes this video, and then we'll continue the rest of the questions in the next video. Thank you very much.